While most spinal cord injuries result in either partial or total paralysis, most of them are actually anatomically incomplete, which means they have spared axons that do span through the damaged spinal segments. Numerous studies have shown that even in the most severe spinal cord injuries, there are some intraspinal networks that can still act as indirect relay pathways to receive and transmit brain-derived motor commands. So the research that we're going to be looking at addresses these questions and attempts to find a way to restart and reignite those axon pathways and connections in the spinal cord to restore some sort of locomotor function.
Using their experimental methodologies, researchers sought to test the effects of CLP-290, a drug that increases expression of KCC2 potassium chloride transporters in neurons, on the functional recovery after partial spinal cord injuries. First, they confirmed that this treatment led to actual target motor neuron activity, not just leg movement during the baso-mouse scale via some other means. Next, to see if CLP-290 was inducing axon regrowth, researchers used tracing methods and surprisingly saw no significant increase in axon development or sprouting for the CLP-290 group compared to the control. They also confirmed that it was the KCC2 expression caused by CLP-290 that was responsible for the functional recovery they saw. Then the researchers found that this treatment had the greatest effect on inhibitory neurons, not excitatory or interneurons, all three of which are found in the spinal cord. And to double check this result, they controllably altered the excitability of inhibitory neurons, and they showed that it resulted in the same functional recovery. This series of experiments led researchers to believe that the downregulation of KCC2 channels in inhibitory neurons was responsible for the major losses of function in spinal cord injury mice. A discovery like this points future research in more targeted directions to improve the functional recovery after spinal cord injuries.
So to summarize the results of this experiment, CLP-290 was determined to be the most successful compound of the ones tested. The majority, which was 80% of the treated mice, recovered consistent hind paw plantar placement and weight-bearing stepping. CLP-290 treatment did not induce functionality in mice with complete lesions, so the spinal cord injuries did have to be anatomically incomplete. So just to restate that, CLP-290 did not affect axon regrowth. However, the compound CLP-290 did show recovery with minimal side effects and promising results in terms of the ability to regain weight-bearing stepping and plantar placement in the rats that were tested. The results of this study offer a lot of hope in future treatment plans for spinal cord injury patients who experience anatomically incomplete injuries, and results do suggest that the reactivation of KCC2 can reestablish a connection between dormant axon pathways and hopefully help patients regain some sort of mobility.